I would be your conductor of the session. Now, I would like to introduce you all to today's speaker, Dr. Kamala Gola, Assistant Professor, Department of Biochemistry and Nutrition, Bhavan's Vivekananda College, who has over 9 years of teaching in various other colleges apart from Bhavan's also, maybe like Pharmacy College, um, Home Science College, Angro, and others. She has done her PhD in JNTU in 2019, and she has also done her MPhil in 2011. She has a very good uh, research background where she has worked in the organizations like Bharat Biotech and also Pharmacy College. And she has also very much interested in am I audible?
Savati, you're audible.
Yes, yes. And ma'am has many papers to her credit, and she has 10 international and 9 national publications to her credit. And I now request Dr. Kamala Gola to please take over the session.
Thank you, Revati ma'am. Good evening, one and all. So, one second, ma'am. I just want to share my screen.
Kamala ma'am, please put
One
your
second.
presentation in full screen mode.
Yes, I'm doing that. Yes.
Yes, it is visible, ma'am. Please go ahead.
Right. Yes, am I audible, students? Good evening, one and all. So now I just want to start this. 1 second. Okay. Yes, BLAST. Previously, I explained nucleotide sequence retrieval from different databases and protein sequence retrieval from different databases in the first class. Are you able to retrieve FASTA sequences from different nucleotide databases, ma? Students, are you able to retrieve the FASTA sequences from different nucleotide databases? Are you able to, ma? Yes.
Yes, ma'am.
Right. So the next step is similar sequence search, BLAST, basic logical alignment search tool. BLAST. Okay. Next level. First, we have to search different databases for different kinds of information, whether it is nucleotide sequence or protein sequence, whatever it may be. We have to search the databases to retrieve the sequences. Right. The next, it is followed by BLAST, basic logical alignment searching tool. Okay, it is developed by Stephen Achu and Samuel Karen in 1990. Okay, so this is basically depending on balance of sensitivity and speed. Okay, it is a very useful tool, right? So what is the uses of this BLAST is, this BLAST is helpful for database similar sequence search. Okay, we are giving something, insulin. Last class, somebody just put an insulin, human insulin. Similarly, they're retrieving another insulin from different origin. Just do the multiple sequence alignment, right? But they are not getting proper results. Okay, so one thing is you have to search similar sequences and pair more than 3 in multiple sequence alignment. Yesterday, that explanation what is done by Dr. Vanitha ma'am, right? But here the thing is, sequence similarities are positive with BLAST. Okay, one known sequence, similar sequences, either 50 or 20, basing on usage of the database, you can retrieve the similar sequences according to your next proceeding level of work. Okay, that is the first use of BLAST. And it is just identify characterized sequences. That means very close, how much close of similar sequences are happened. That information is also given in the results. Okay, and phylogenetically how much the sequences are related. BLAST is providing first thing similarity and characterization of the sequences, and phylogenetically how much they are related. And we can also use this tool to identify possible functional based on. Okay, so BLAST can helpful for identifying similar sequences and characterization of sequences, phylogenetically how they are correlated with each other. These kind of informations we can get for a unknown sequence or known sequence, which is pasted in the search blog. Later I will explain the. Okay, so this program, BLAST program, see here usually different kinds of BLAST search are possible in NCBI and different databases, right? So BLAST N, BLAST N, BLAST P, BLAST X, T BLAST N, T BLAST X. These are all are possible in NCBI. Okay, so BLAST N means database subject is nucleotide. Okay, so I will explain one by one, right? So this is BLAST N. The query is nucleotide sequence. The query is nucleoside sequence, and the result also nucleotide data sequence. Similar sequences executed by using this program, we are getting a nucleotide bases which are very similar to the query sequence. Are you able to follow this one, ma? BLAST N means nucleotide sequences. We just keep one nucleotide sequence in the search engine in the FASTA format, and the results we can expect it. That is also a nucleotide sequences. Is it clear? BLAST N is, yes ma. Are you able to follow this one? BLAST N is helpful for searching the nucleotide sequence. Query is nucleotide sequence and the result also nucleotide sequence. Is it clear? Yes, students. BLAST N, N for nucleotides. Okay, query sequence is nucleotide sequence and the results are similar nucleotide sequences. I will explain with one worksheet. Yes, ma. NCBI. NCBI, popular resources, right? At this corner, BLAST is there, right? You just highlight the BLAST, right? So in this website, we can able to do the nucleotide BLAST, nucleotide to nucleotide sequences, or protein BLAST, protein to protein. That means your query sequence is protein and the resultant similar sequences are also proteins. Is it clear? Yes, ma'am. Are you able to follow this one?
Continue, Kamala.
Yes, nucleotide blast. Okay, so now I just go with the nucleotide blast or blast N. Is it clear, ma? So now I just want to take one faster sequence. Here I am keeping insulin, Homo sapien insulin. I'm getting faster. Okay, I just copy this sequence and paste here. Asta sequence I pasted here. And next, blast. Okay, so see, these many similar sequences are executed with one query, right? So now you just check what we are getting here. So the first one is the our sequence, and second one is the first one is the query sequence, and the second one is the sequence what exactly with the INS IGF2 sequence. This is located on chromosome number 11, right? So similarly, those sequence which are very similar. Okay, they are just grouped in this way, right? So now how much similarity is there? So percentage of identity, I see identical similar sequences with this. You have to check 100%. That means query sequence and the resultant sequence have a similarity of 100%. Is it clear, ma? Is it clear? Is it clear? Yes, ma. Students respond. Yes, students.
Kamala ma'am, they're responding in the chat box, so you can continue, ma'am.
Okay, right. So similar sequences are displayed on the result box, right? First, we have to take the query sequence and get the result sequence, right? We can download all the sequences at a time, or we required only 10. We just restricted to this 100. We restricted to either 10 or either 50, either 100, right? So in this way, you have to select the options, and accordingly we required faster sequences of all these resulted sequences we can download a test stretch. Okay, so here one thing, once you just see this result, see our query sequence is nucleotide sequence. Okay, resultant sequence is also a nucleotide sequence. Is it clear? Our query sequence is nucleotide sequence and the resulting sequence is also nucleotide sequence. This kind of results are happened in BLAST-N. Okay, ma? Right. Next one is BLAST-P. BLAST-P means here the query sequence is amino acid sequence. The resultant sequence is also amino acid sequence. Okay. So the query sequence is amino acid sequence and the resultant sequence is also amino acid sequence. That is the reason BLAST-P. Upon searching of BLAST-P, we can search similar polypeptide chains or similar proteins. Right. See, in this workplace, BLAST-N, we already used one gene sequence and we just search for similar sequences. Next, I just move on to BLAST-P. That means here the input must be a amino acid sequence. Is it clear? I just retrieve this amino acid sequence from NCBA. So here I am highlighting protein. Again, I am keeping insulin, Homo sapien insulin. Search. FASTA sequence. This is again it is taking me period. See. Now I'm taking the amino acid sequence. I'm keeping this sequence into this query box. Right. Now I just select the BLAST-P, protein BLAST. Okay. So this is the result. Right. Previously, we just go with the BLAST-N, similar nucleotide sequence we can get. Okay. But BLAST-P, amino acid sequence we are pasting in the query region or query box. Now we are getting similar amino acid sequences. We can check. See, ma, this is the query and this is the result. How much similarities are there? 100% similarities are there. So every sequence is aligned with the query sequence. How much similarities are there? Basing on that, how much correlation is there? Percentage of correlation between these two sequences. It is displayed as 100% or 90% accordingly. Okay. So we can download the entire 100 sequences or 50 sequences, but here amino acid sequence. That is the difference between BLAST-N and BLAST-P. Are you able to differentiate between blast N and blast P, students? Just give me the quick response. Yes, students. Blast N for similar sequence search with nucleotides. Okay. Blast P means similar sequence search for proteins. Okay. Now I just move on to the next. Madam, my PPT is visible.
Yes, Kamala.
Okay.
You're
Right.
sharing the NCBI screen, no?
No, ma'am, PPT. That is the reason I'm asking.
PPT is not there. NCBI screen is.
Still it is there now. Okay, Nisha. I would like to share the. Yes. Right. So now we complete the blast P and blast N. Now we just move on to next. Blast X. So here the query sequence is nucleotide sequence, but the database provide the result is amino acid database. Okay. So here this is a different thing, a translated frame. Here the query sequence is translated towards to amino acid database. This is a different thing. Usually we just use the blast N and blast P. Okay. Blast X. Next one, T blast N. Here amino acid sequence is the query sequence and the output is nucleotide database. Right. Next one is T blast X. Here the query sequence is nucleotide sequence and the database is also providing nucleotide base sequence, but it is 6 frames are translated. Okay, that is the difference between blast N and blast P. Blast X, T blast, one second, T blast N, T blast X, right? So now I just explained this algorithm, right? Basic strategy involved in blast algorithm, right? So the query sequence, whatever the sequence we posted in the query box, right? The similar sequences are expressed in the display. Okay, what exactly happened? Direct match. How much similarities and how in NCBI, how many segments are similar to the required query sequence? Usually 12 similar score, see, 11 characters and the score should be 12. More than 12 resulted in blast P. Okay, so it is completely depending on Blossom's 62 matrix. Okay, an algorithm which is calculated how many, see, you are posting 100 sequences, 100 words or 100 letters, not words, 100 letters, right? So 100 letters, whether completely similar sequence only identified. So the here algorithm is completely different and it is proposed to similar words which are present in the query sequence as well as result and target sequences, right? So both the sequences are simultaneously executed. Then only it is giving the similar sequence approach with respect to blast P and blast N. Yes, ma'am? Yes, are you able to follow this one? Okay, so now I just explain with the blast P. Okay, So now I just take the 6 alphabets. I'm not going with any. These are alphabets which are similar to amino acids, single letter codes, right? So every single letter code is responsible for 3 nucleotide base sequences. First, I go with the BLAST P, right? So this is, see, this 6 is aligned in 4 different patterns, right? So in this alignment, maximum possibility score must be more above 10. Those sequences only added and those are displayed in the summary sheet. Are you able to? Query sequence and targeted sequences. Whatever the query sequence which has pasted in the BLAST page, right? Then immediately simultaneously it is summarize the thing and display the target sequences. There is a minimum correlation. That means 10 sequence must be correlated with the query sequence. Those kind of sequences only explained are resulted in the similar sequence search result portal. Yes, ma'am. Are you able to follow this one? Yes. Are you able to follow this one? So minimum of minimum length of the fragment is 12. 12 pages or 12 words or 12 characters. Similar sequences are there. Then only the sequences are aligned according to their similarities, right? So here 10 to 10 is base pairs are there. It is 100% similar. That is going to be the first target sequence aligned in the BLAST result display paper or display report. Is it clear? This is again explained by S. Padma ma'am. It purely algorithm is explained by S. Padma ma'am. Yes, ma'am? Why do we take baseline 10? Right. I'll explain. 1 second. Default threshold. Okay. So NCBI BLAST P use a default threshold 10. Okay. That is the reason we consider 10 with minimum of 2 gaps where that score is denoted as 12. Is it clear? Yes, ma'am. Okay, right. Now we just move on to next. Okay, so this is the overall information. 1 second. Just go with the overall types of blasts. How can we get the outputs, right? So blast N, I already explained with NCBI. So nucleic acid sequence we are using as a query sequence and the output is also nucleic acids. Is it clear? Then peptides or proteins are the query sequence. Resulting thing is peptides. Okay, but T blast X, are you able to see this one? Blast X, we are giving a protein translation. That means nucleic acids. Here the input is nucleic acids, the output is protein. Here translate we are able to carry forward with this BLAST. Translate nucleic acids to protein. Similar type of proteins are executed. If you are keeping nucleotide, the executing files are proteins. Okay, next one, TN BLAST X, TN BLAST X, right? Here the thing is nucleic acids translated to protein and again protein translation. See, there is a nucleic acid to nucleic acid with the co-intervention of proteins. Is it clear, ma? Nucleic acids to proteins, but here the protein translation is going to be happen in BLAST X. T BLAST X means input is nucleic acids and it undergoes protein translation and result the protein. Is it clear? Okay, T BLAST N is peptide or protein is the input. Okay, and here nucleic acid is the output. Is it clear? Is it clear, ma? Are you able to differentiate all these things?
Yes, ma'am.
Right. You can use SEMA. You just work in NCBI BLAST site with all tools available. I just share the screen. 1 second. I'll share the screen. Okay, right. So SEMA, BLAST N, BLAST P, BLAST X, T BLAST N, T BLAST X are possible, right? So you just paste the FASTA sequences retrieved from the NCBI. What kind of output we are getting, right? You just check, then you get a clear-cut idea. Okay, why we are always taking a protein? If we are doing a nucleotide search also, we are just typing a protein molecule. Why means protein having less number of nucleotides. Okay, but if we go with the genome, more number of gene sequences are there, so we unable to complete the results and we can't execute that files immediately. That is the reason we, even though nucleotide search also, we are using a insulin or a small proteins. Is it clear, ma? Are you getting the clarity with the NCBI BLAST? Yes. Yes, ma. Are you able to follow this one? Please do little bit of exercise. Just go with the nucleotide sequence in BLAST N and amino acid sequence with BLAST P and nucleic acid sequence with BLAST X, T BLAST N, T BLAST X. Please do try with the different sequences. Is it clear? Is it clear, ma? By practice only we can get the clarity. Okay? Right. Now I just want to explain the different databases. Different databases, right? So gene banks or gene bank NR. It is a database which is consisting of proteins and nucleic acids. Okay, both are available in this. So you can retrieve the FASTA sequences and do your work in NCBI BLAST. Okay, so these databases can directly run the BLAST. Okay, so NCBI BLAST service is available in NCBI, right? Apart from that, so many BLAST tools are available in the internet. Okay, so first priority goes to NCBI BLAST, right? We can do the nucleotide BLAST as well as protein BLAST with this. Okay, next Uniport BLAST. It is the another one. See, I just explained the list of the BLAST tools, right? 6 different tools are available in the internet, right? So the first one is NCBI BLAST. So maximum work we can complete with the NCBI BLAST. Next one is Uniport BLAST. Okay. So now I just want to move to Uniport BLAST. See, ma'am, just check with the Google, right? Uniport. Uniport BLAST is there, right? So you have to use the Uniport ID, or again you can use the sequence, either protein sequence or nucleic acid sequence. The sequence must be in FASTA format. Remember, guys. See, you just use number of online tools, right? But be clear, Uniport, whatever work we carry forward in Uniport, you must paste the FASTA format of respective sequence, whether you are working with nucleotide sequence or protein sequence. Is it clear, ma'am? So now I just go with the protein sequence. Okay, I just paste the protein sequence. Okay, right. So here that is a protein and BLAST P SEMA sequence type, either protein or DNA RNA. Next, I have to choose the protein. Why means I just paste the FASTA sequence of insulin protein, right? So which one is required? BLAST P or BLAST X? P alphabet for protein, right? Threshold 10, right? Matrix. Okay, so this how many hits we required? We have to minimize to 50. We just remove the hits. We can reduce the time interval to produce the results. That is the reason. Okay, so now I just run the BLAST. Okay, I didn't enter the email ID. That is the reason my job is failed here. Right. Why there is no portal here? There is no. The server is not accepting the. So some issues. Padma ma'am?
This Kamala?
Ma'am, I'm not getting this results, ma'am, in Uniport. Previously, I carry forward this one, I get the results.
You open that, this is in you open the Google, this thing. This is your PPT, right? You open in the browser.
Browser only, ma'am. I'm opening in the
PPT
browser.
showing. PPT showing.
Okay, I just to go with the browser only. Yes, ma'am. Here I'm pasting the sequence.
Go up.
Okay. Yes, ma'am.
Go up.
If I'm running this one, job is failing. Why I am not getting? The server has not.
Job is running, no? It is in the queue, I think.
No, ma'am. Previously I failed C. I'm getting C. Failed.
No, go back. Go back.
Yes, ma'am.
Here it is not. Did you paste the what sequence you have pasted? Once
Faster
again, you
sequence,
paste.
which is retrieving from the NCBI. So this sequence I just took from this NCBI site.
You give the Uniprot ID there. P01308.
P01.
Remove the sequence here.
8.
P capital P. Capital P01308.
Yes, ma'am.
Remove the sequence down.
Yes, ma'am.
Now
Right.
you try. Now you try.
Actually, okay. Failed, ma'am.
Something else you have.
By 4:30, I did the same job. I am getting the results.
It's okay. We'll try later.
Okay.
Let me check.
Okay, ma'am.
You carry on with your class. I will check and
Yes,
get
yes,
back.
ma'am. Yes, yes. Yes, students. So we can use different tools which are available online and we can get the similar sequences. Okay. We can get the similar sequences. Okay. First one is NCBI. Next, Uniport. Right. Uniport is not giving proper results. Just you check, ma. This is easy. We can get the similar sequences. Okay. Right. Next, another one is this is Quadral Lab is providing one online tool. By using of this also, we can get the similar sequences. Okay. Next online tool, bio tool. Right. So this is the fourth version of blast tool, right? It is available in online. We can paste the sequences, faster sequences. Yes, please mute yourself, ma. Please, somebody please mute yourself. Okay, so this bio tool is also is a tool available in online. We can do the use the faster sequence for blast. Similar sequence search is possible. It is a online tool. Is it clear, ma? Right? See, these are the 6 different tools, links. Okay, we just use all these links through internet. We can check the blast through online. I'm pasting that, right? So this is EMBL, EBI. You already use Clustall W and all, right? So find, we can just check the similar sequences by using this database. Is it clear, ma? This link is also providing similar sequence search. Is it clear? Are you able to follow this one? Yes, students. Are you able to follow this one? Nobody. Please respond, students.
Yes, ma'am.
So all together, ma, first thing is NCBI are different databases. I explained 6 different databases to retrieve the sequences, right? You are working with different primers and different sequences, right? Different gene sequences, different proteins, right? So what is the use of BLAST? What is the importance of BLAST? Okay, BLAST means we are using one query sequence, similar kind of sequences which are depository in the databases. They can easily retrieve. Okay, so after retrieval of things, tomorrow I will explain the generate 2D structures and 3D structures, structural similarities. We can find out with the help of this BLAST results. Is it clear, ma'am? So after getting of BLAST, we just align the sequence with the help of multiple sequence alignment tools. Clustal W and Clustal Omega, already explained by Dr. S. Vanitha, ma'am. Is it clear, ma'am? So multiple sequence alignment is feasible after knowing of your sequence and simultaneously similar sequences, whether it is 10 or 50 or 100. It must be more than 3, remember. Okay, so after doing of multiple sequence alignment, unknown structure, unknown sequence, that means you are generating one sequence. Recombinantly, we are generating one sequence, but we don't know the 3D structure of that protein, right? So we can using all these query sequences already deposited in the NCBI database, and we can generate your protein structure. Are you getting clarity? Any drug discovery is happened with the help of a protein molecule. That means this protein molecule is bind with the drug component or is showing any interaction. First, we required a protein structure, right? So by knowing of the proteins, we just go and check the NCBI database. Okay, so if NCBI database having the protein, we can get the 3D structure. There is no issue. But you are searching a protein which is not having 3D structure, those kind of proteins. Okay, first you have a FASTA sequence. Just go and paste FASTA sequence in the BLAST portal. Right. Similar sequences are resulted. Right. So 100% similar sequence, 3D structure is there. So you have to use these two sequences furtherly and retrieve the structure of unknown sequence or query sequence, what exactly you pasted in the NCBI database. Is it clear? Are you getting clarity? How to get the protein structure? Later I will explain. Okay, next class on Monday, I will explain the structure development with the query sequence and target sequence. Is it clear, ma? Are you able to follow this one?
Yes, ma'am.
Are you able to do the BLAST, ma? Even nucleotide sequences and even protein sequences. Basically, 2 sequences are most important. Okay, transcript is little bit tricky, right? Transcripts plays a major role with respect to coding sequences and non-coding sequences. Okay, so basic thing is fundamental thing, nucleotide to nucleotide sequence alignment and protein to protein sequence alignment. NCBI BLAST is the best one. Best online tool we can use in multiple ways. Apart from that, 6 online tools already I just projected in this PPT. We can use those things and we can get the similar sequence search. Is it clear?
Yes, ma'am.
Yes, ma'am. So anybody having any doubts regarding this topic? BLAST.
Can you explain once again T-blast and T-blast X?
Sorry, ma. Sorry, I'm not getting.
Can you explain once again T-blast and T-blast X?
T.
T-blast and T-blast X.
Okay, T blast and T blast X. Yes, I'll show it to you and I'll tell you. Okay, ma. Are you able to see the screen? Seema, T blast N. Okay, the query sequence is amino acid sequence. The database giving an output is nucleotide database. Are you getting clarity?
Yes, ma'am.
So here our query sequence, Seema, translated. Frame 6, all 6 frames are translated in the database and searched with the protein sequence. Right? That means most prominent genome mapping in between protein and the respective genome is going to be happened in T blast N. Is it clear?
Ma'am, your screen is not visible, ma'am.
Screen is not visible. Huh?
Yes,
It is
ma'am.
sharing, ma. It is sharing. See, ma. One second.
Ma'am has shared the PPT.
I'm sharing
I'm
the
able
PPT.
to. Yeah, I saw just now.
Ayyo, one second.
Malli poyindi Kamala.
Yes, ma'am. Now?
Yeah, it's visible. Mahesh, it must be visible to you.
Yes, ma. Are you able to see the screen?
Just check your net. That's it. Nothing else.
T blast N means the query sequence is amino acid sequence. The output sequence is nucleotide sequence. What exactly happened here? Frames are translated and searched with the protein sequence. There is a mapping between protein to a genome. That is nothing but T blast N. Is it clear, Mahesh? Yes. Next one is T blast X. The query sequence is nucleotide sequence.
Ma'am?
Okay. Yes, ma'am. Are you able to follow this one?
Yes, ma'am. But
T blast N.
yes, ma'am. If you take the nucleotide sequence in the T blast N, ma'am, what will happen in the result? Ma'am?
No. There is no output. See, T blast N. The query sequence must be amino acid sequence. You should post the amino acid sequence, FASTA sequence. Then you get the output, which is a true translate of that amino acid. So that is the reason, see, mapping of a protein to genome. Protein to genome. This technique T blast N is a tool which is helpful for the to translated protein with the genome. Is it clear? That means that protein is going to be RNA sequence, then furtherly develop the DNA. So nucleotide data sequences resulted. We can check with the NCBI blastma. We just post the amino acid sequence, then you can get the results. If you are posting nucleotide sequence, we can't get the proper results. We can try with the 2 sequences, no issue. Okay?
Okay, ma'am.
But the
Okay.
query sequence is amino acid sequence in T blast N. Okay? Got it up?
Yes, ma'am. Yes, ma'am.
Right. Next, T blast X. T blast X. This is the most important thing. The query sequence is nucleotide sequence. The database sequence is nucleotide sequence. Okay. The 6 frames are translated on query on the database. Searching distinctly related species. This is the most important and at most different kind of blast. Here, searching this blast is helpful for distinctly related species. Okay? Distinctly related species we can search with this. What exactly happened? What is the translation? Nucleotide sequence is the query sequence and it is translated to protein, again executed nucleotide base sequence. That is the beauty of T blast X. Is it clear, ma'am? See,
Yes, ma'am.
I just explained this. See, ma'am. T blast X. See nucleotide sequence, and here also nucleotide sequence, right? So this input query sequence is a nucleotide sequence, and it is translated. Okay, similar type of protein is searched, and it is again displayed the database in as with nucleotides. Are you getting clarity? T blast X. Okay, this is not similar to blast N. Blast N, what exactly happened? Similar nucleotide sequence is there that should be displayed in the display board, right? But here what happened? T blast X. So nucleotides are converted into protein translates, sex protein translates, and similar protein translates are checked, and next they are converted into nucleic acids. Is it clear? Are you able to follow this one?
Yes, ma'am.
Okay, so these are the different blasts available in NCBI. Another tools also you can try. I just post the different blast tools available in the net. Okay, now I just want to post this 6, the chat box. Okay, you use any of these things to get the blast. Okay, any queries, ma? Any questions? Yes,
Students, if you have any other queries, you please come up with.
are you able to follow this class, today's class? Blast. Still, do you have any doubts? How many people able to follow today's class? Just respond in chat box. How many are able to follow today's blast class? Respond in the chat box. Yes. Okay. Thank you very much. Right. Padma ma'am? Revati ma'am? Shall I wind up the
Yes,
class,
ma'am.
ma'am?
Okay.
Okay, ma'am. Thank you, students. Revati
I
ma'am.
hope there are no other queries, students. We are moving on to our next lecture, next session. Dr. S. Padma ma'am would be taking on to this session. I hand over the session to Padma ma'am.
Kamala, you have to stop sharing, I think. Is this screen
Yes, ma'am.
showing?
Yes, ma'am. Yes, ma'am. Yes, ma'am. Okay, na, ma'am? Now it is okay?
Yes, Kamala.
Okay. Thank you, ma'am. Thank you.
Revati, thank you. Okay, students, so now that you have gone through the blast, that is alignment of sequences, here next I would like to add a few more um, sentences regarding the blast alignment. So generally, whenever here in the previous section, Dr. Kamala had told you how to convert a nucleotide, how to compare a nucleotide sequence with a protein and a protein to protein or nucleotide to nucleotide blast. So when we do a blast alignment, we are trying to see the extent of similarity of sequences. So here, one more I will add on to the same thing, what continuation of what was discussed by the previous speaker. So here I will just show you another process. Let me just share the screen. Just only for the purpose of continuity. I'll just open the window and then share this. Yeah, I'll just share the screen now.
Visible, ma'am.
Not this one. I wanted to
Yeah, it's
share.
predicting RNA structure.
I wanted to show NCBI side.
Yes, ma'am.
Okay. So here,
The screen is visible, ma'am.
okay. So students, you all are now well versed with this site. So here we can take any example of a sequence. This is just an add-on to what you have gone through just in the last session. So this, I'm just selecting some sequence here, an mRNA sequence or whatever it may be. Okay, and then what you need to do is go to your browser and do another search which is called as a Needleman Wunsch. Just you type this particular one that is called as needle pairwise alignment. Okay, so this is another add-on to the sequences or whatever we have done. So here, this is an emboss needle. This is why it is called as emboss and why that needle is because emboss is the trademark like your EMBL site that is called as emboss and needle is a name which is given to the algorithm based on the algorithm which was uh, proposed by two scientists by name Needleman and Wunsch. How many of you are aware of this, students? Anyone is aware of this? Any of the participants are aware of this? Participants, are you aware of this? If you are aware, then we will go further. Otherwise, I will go through this.
No, ma'am.
No? Hello?
No, ma'am.
Okay. So, this is a type of tool like you T-Blast 10 and other resources. You can also use this when you want to just compare two sequences, T-Blast 10 or when you want to compare the protein to protein or nucleotide to nucleotide, what was shown in the previous session. So here also you have algorithm is nothing but the calculation which goes behind the program, how to calculate the sequences, how to compare the sequences. What we are trying to do here essentially is if you are identified a sequence or you have done a DNA sequence of some species which you have isolated or which you have done and you want to see whether such kind of sequence is already available through the databases, then you can do this pairwise of alignment. So if you are using a DNA sequence, you need to select the DNA sequence and then you have to paste your first sequence here and paste your second sequence. So here again, you need to paste the FASTA sequence into this particular window. So how do you get the FASTA sequence? You know you are well versed with it. Go to your browser, go to the NCBI site and take the molecule of your choice, nucleotide of your choice. I'm comparing the mRNA sequence. Why I'm taking mRNA is because if I take the genomic DNA, the size is too large. So many base pairs are there. You need to search whether there is a smaller one. shorter DNA sequence we have done earlier. You can see here there is 1600 base 1660 base pair linear DNA is there, and then this 1206 base pair linear DNA is there. You can use these sequences, or you can use an mRNA sequence. Yesterday, you were told in the class about these mRNA sequences also. So this again, we need to take the FASTA sequence as we do with the earlier any alignment process. So you can copy the sequence and put it on to your alignment here, and this is the same process you must have gone through in the previous session also. Since I was not going through the complete session, I was not there. So if you are done already, just you do the same thing here. So you have to take the FASTA sequence and you need to copy and you need to put it back here, and then you need to submit. So so what is the purpose why I am showing this is one of the very basic techniques or basic tools when you are trying to do a blast alignment of sequences. So this is called the result, which is shown here. You can open this sequence, or you can look the pairwise alignment, and you can take all these results into your folder and visualize the results. So output is given here. You can see the Needleman-Wunsch algorithm. This is showing some terminology here. How much of identity is there between the two sequences? How much of similarity is there between the two sequences? And what is the score and what are the gaps? What is this identity? What is the difference between identity and similarity? So if you are going through these residues, you can see here, these two are the accession numbers of your sequences which you have taken. These are the query sequences. And here, because already we know that the base pair length is different between the two. When we took the sequences itself, we have taken some longer and shorter sequences. So here, you can see the alignment is starting from this position. Now, what I want you to understand here, because whenever you are doing a sequence alignment between two sequences, you have to observe what is the position of the alignment which is done. Okay, so now you have to why this is important is there are two types of alignment. One is this alignment which we call it as the global alignment of sequences. Global alignment. What is meant by global alignment of sequences? Global alignment is matching the sequence to sequence. All the residues are going to be matched. Okay, so here if you see here, this is starting all these nucleotide positions. There are some, this is one sequence and the second sequence, because it is a pairwise alignment. If you recollect your clustral alignment, in clustral alignment we did not see this kind of bars. Why? Because it is a multiple sequence alignment. Here it is a pairwise sequence alignment, which is looking at what are the individual nucleotides which are correctly matching between the two sequences which we are taking. So you will be observing that at some places there are some gaps where there is no kind of similarity in this output. So here you can you are not finding like your clustral alignment, you are not finding any stars or anything here. You are only finding the bars. Wherever you observe the bars, you can see that here the two sequences are missing. So two nucleotides are missing. So this kind of calculation is possible here, which is called as a gap penalty. Why gap penalty? Because when you are comparing two sequences, there is a gap which is coming up, and the score here you can see you are giving a score to your sequences, which means that you are giving some marks if they are matching, and you are removing the marks if they are not matching. If there is some space between the two sequence or a gap between the nucleotides, then you are deducting some marks for that particular sequence alignment. So this is the basic technique. After later you will also this first, if you understand why this scoring is coming, what is importance of the scoring, later you will be comparing a nucleotide with a protein or a translated sequence with a protein sequence and so on. But this forms the basis where you will be seeing that the identity and similarity are here. You remember this value, remember this figure, or you can take a picture or whatever it is. Let us do in another source another type of alignment. This is one alignment which we have seen. So now let me show you another example. Yes.
Ma'am, can we discuss the Needleman-Wunsch algorithm in detail? I guess
Yeah.
that's what we are seeing right now, the results of that.
Yeah, if you have to, what is the detail you would like to have? Because it itself will take some time. I have to explain to you what is gap penalty, what is all this, how the scoring is done. It will take little time. What is it that you would like to know?
Ma'am, actually, I wanted to know, like, how is this working? Like, how are we calculating the gap penalty and all this?
Gap penalty means, suppose here, because I don't have, I'll just share the PowerPoint and let me show you. Give me a little time.
Yes, ma'am.
Let me share my slides. I will just put one, I will just put one slide and on this window itself and show you what it is. Okay, so now because I have no pen attached to this, I'm just trying to show, so you have to bear with me for this handwriting. So I'm putting 4 sequences here. Some random sequence I'm putting A, G, C, C, T, A, like this. I'm putting some sequence here and T. This is first sequence 1. This is sequence 1 for me. Then I'm taking the sequence 2. Just you have to, because I'm trying to use the mouse, so just bear it with me. So I'm putting one A here. I'm just randomly taking a sequence. I'm not putting G here. I am putting a C here. I am putting a T here. And I am putting an A. So these are not complementary sequences. Remind you that these are not complementary sequences. We are taking a sequence 1 from one source and sequence 2 from another source. Is it clear? So we are not taking a complementary sequence, so don't expect that it would be complementary, first thing. So when you are doing this kind of alignment, you have a gap here, because how many positions are there here? 1, 2, 3, 4, 5, 6, 7. And here there is 1, 2, 3, 4, 5, 6. Is it not? So here there is a gap in this particular sequence. In this you have how many bases? 7 bases. And in this you have how many? 6. Is it not? So
Yes, ma'am.
what the algorithm does is it is going to give some marks for this, like it is going to give a match. If this is matching with this, it will be giving it a score of 1. If there is a gap, this is going to be given a gap score of -1. Okay, that is, it is giving some penalty points for that sequence because there is no matching for that particular nucleotide. And here it is matching, but so you are getting 1. Here it is matching, it is not, it is mismatching, so it will be given as 0. Score is not given here, score is given as 0. Like here again you have a mismatch score is given as 0. Here you have a score of 1 and here you have a score of 1. So totally how much score is there here? 1 and 1, this plus 1 minus 1 is cancelled, and totally you have a score of 3 for this, right? So this is one way of writing the sequence. The same two sequences can be written with the gap somewhere else. This is the gap I am creating. Can put a gap here, or I can put a gap here, or I can put a gap in anywhere of the other positions to make sure that I am getting the position at 5 different locations. So every time I generate a sequence, it is giving a different kind of score, which will depend upon the match points and the mismatch points. Did you get?
Yes, ma'am.
Yeah. So
Ma'am, but if every time the alignment is changing, then how are we
yes,
to
yes.
administer
Yes,
the
if every time the alignment is changing, what is our what is the goal of our no, what is our goal? Our goal is to see which is the best alignment possible. Is it not? Now, if I go back to my slide, if I go back to this site, I'm going back to this. So you are seeing the screen, right? So,
yes,
so here, what is the reason that only this alignment we are accepting? Why are we accepting only this alignment? Why it cannot start from here? Why it cannot start from any other place? So the reason here, it is based on these scores, based on these values which are getting the least this alignments, after deducting the gap penalties, after deducting the match score, after adding the match score, removing the mismatch score, you may get values like +3, -3, +2, -2, like that. So the one which is having the maximum score is the one which is the correct alignment. Is it clear? So now we cannot be doing for 5 nucleotides, you can write comfortably, you can be putting it on 5 different sequences, each time changing the position of a nucleotide. But if you are looking at now the 2000 base pairs or 2000 nucleotides, you cannot imagine how many combinations you could write in that. It is like, if you remember, construction of phylogenetic trees, where I showed you that 2 to the power of n number of trees are possible for a large number of sequences. So in the same way, when you are comparing different types of sequences, it is it is not possible, or it is not in our constraint that we can do it manually. So we take the help of some mathematical program, like the dynamic programming method. So the dynamic programming method will use a matrix. So if I start explaining that, this whole class will be sufficient only for that. So take my word, dynamic programming matrix will be constructed. You first have to understand what is that dynamic matrix, dynamic programming. So this particular algorithm, that is the Needleman-Wunsch algorithm, is based on the dynamic programming method. So just to make you understand, I will just show you in this particular window what this dynamic programming means, make you understand. Or later I'll show a slide at the end of the class, which I have earlier slide. So what it does, is it will be constructing it will make a grid like this. There will be a grid which is constructed, and in this grid there will be some rows like this. Okay, all the rows will be lined up like this, and you have some columns which are there. Okay, you have some columns which are marked on this, and this dynamic programming method will put all the sequences on this axis, your sequence 1 on this axis, and the sequence 2 on this axis, like this. Okay, and it will initiate this with multiples of the gap penalty. So there will be one more row where it will be initiating with gap penalty. So here it may be, let me just put it erase this. And so here I am putting one more line. So where it is going to be initiating with a gap penalty. So this is a completely mathematical calculation, starts from 0, and it would have my multiples of gap penalty -1, -2. It goes on like that. Whenever there is a my gap penalty, the value is minus. So the values again, the sequence, one sequence which is going to be matched is going to be put on this end, and the other sequence is going to be put on the y-axis or x-axis like this. And this calculation will be based on the dynamic programming matrix calculation. At the end of the calculation, this grid is going to be completely filled with numbers, and then the sequence is written in such a way that it goes from here and reaches to the top of the table. It goes in a diagonal manner. So during this process, it sees that all these values which are following this line, these are in the descending order. So it starts, suppose this starts from 4, it will be 3 here, 2 here, 1. Again, if it is 1, this will just move like this. So based on that, you would be getting the sequence. Later after my session, I will also, if time permits, or if you are still going to stay back, I can just show you later. Is it clear?
Yes, ma'am. Understood. Thank you so much, ma'am.
Yeah. So later, if you are still there, I will show you with another slide of mine. Okay. Anyone is raising
ma'am.
hands? Someone is raising hands?
Mahesh has raised his hand.
Yeah, Mahesh, tell me.
Nothing, ma'am.
Okay. So that's how this particular alignment will come. Now similarly, so this is called as the global alignment of sequence. Now we will also see another alignment which is called as the Smith-Waterman algorithm. Okay, so Smith-Waterman algorithm, this is also a local alignment of sequences. You can see this one, emboss water. Emboss water is also another way. Previously, we have seen emboss needle, is it not? I have shown you the previous one with emboss needle. So this is
Yes, ma'am.
emboss water. So what is the difference between that alignment and this alignment? When you see it, you will appreciate. So again, we'll go back to our NCBI site. So you can, I will go back here. One, yeah. So what are the sequences I have taken earlier? I think I have taken this one. I don't remember. Maybe this one is 1660. I will take this sequence. I'm going to put it over here. And right, so again here you can put the sequence and you can submit. So now Did we take the same one? I don't know. Whether I took the same numbers here. You have the screen with you have taken earlier picture, you can see. Okay, now you can see here. Now in the previous alignment, what is it that we have got? If you have the alignment or you have taken the screenshot or whatever, I can't see here. Now you can see the identity similarity here. Here it is showing 48%. What is the difference you are observing in the alignment process? In the previous alignment, you have seen that the alignment is starting at some other place where the first sequence of one nucleotide with the first sequence of the another nucleotide, another sequence. Here you can see that it is not focusing on the first base, but it is focusing on which is the nucleotide of this sequence that is aligning with the nucleotide of the other sequence in the pair. So it starts to align from that position. So you can see here for this sequence from the 9th position, the alignment has started. For this sequence, the alignment has started from the 3rd nucleotide position. So what is the purpose of this? This is called local alignment. What is meant by local alignment? Local alignment is it is trying to see what is the best way it can compare the sequences so that if any conserved sequences are there or conserved regions are there which are comparable between the two sequences, these are not missed. So that is the idea of doing a global alignment and local alignment. If you come, you can sit and do it for yourself and put both the alignments next to each other and you can see what is the difference. Where you will find the difference is in the extent of identity of similarity and the gaps you may be finding in a difference there, and the number of gaps also may be different. Is it clear? Yes? Yeah. So that is what I just wanted to add on to the other lecture by Dr. Kamala, where you that is a step where you are going to compare the proteins to proteins. There itself, other varieties of blast are there. So blast, why we have the blast P or blast nucleotide, T blast N, T blast X. So these are when you have a type of sequence on hand and you want to convert it into a nucleotide or you want to convert it into some other format, those blast formats can be used. But this alignment of sequence is only between a pair of sequences. So you take two nucleotide sequences or two protein sequences. But if you want to see how much of the protein sequences are coming from the nucleotide, then you can see the possible frames which will be coming up. What are all the types of possible sequences which will come if you take a transcript or if you want to convert a nucleotide into a protein, how many possible combinations will come up. But if you are only interested to compare a sequence 1 to sequence 2 and you would like to see, I have taken a worm sequence, I have taken a fly sequence, I want to see similar between the worm and the fly, then this kind of alignment can be done to show sequence similarity or the gaps with the gene of interest. Okay?
Yes, ma'am, understood.
Yeah. Okay, so that's for the sequence similarity. Along with the other types of blast, we should also learn to use the algorithm and do the calculation. Now let's move on to the other topic of today.
Ma'am?
That is, yeah.
Ma'am, before that, can you please discuss one or two like applications or examples of where local alignment can be used?
No, there is no example like that. You can use local alignment for any of the sequence. You take any two sequences of your interest, you will use a local alignment. Okay, global alignment is you can compare both global and local alignment. There will be some difference with respect to the alignment of sequences. This global alignment and local alignment is actually BLAST. Global alignment is only an alignment of sequences. It is not a part of the BLAST. Make it clear.
Yes, ma'am, understood.
Yeah. So if you see BLAST, let me see BLAST. BLAST will be local alignment sequence tool. If you are using BLAST, you are doing local alignment. Okay. If so, look, that will be based on your Smith-Waterman algorithm. If you are using global alignment, you will be doing it not as part of BLAST, but you will be doing it separately. Okay. Shall we go further? Okay. So let's move on to the other topic. That is, I'll just share my screen.
I think you have to share it now, ma'am.
Yeah, I will share. Okay. Is it sharing? Yeah.
Yes, ma'am.
Yes. Okay. So in the last class, in yesterday's class, Dr. Manju Devi had explained to you RNA structure prediction and how to predict the structure of an mRNA, how to predict the structure of tRNA, and so on. We have used different databases, different resources, different tools to predict the structure of RNA. And you were also explained about the significance of the RNA work, why RNA is important, because it is the transcriptome of the cell. The transcriptome is going to be deciding what are the proteins which are going to come out. So from genome, you are getting a transcriptome. When you get a transcriptome, it is mostly containing the exons, and then the transcriptome will get translated into the protein. So as how important genomics is, the transcriptomics is also equally important. So you have been told that there are different types of RNA along the coding and the non-coding RNA. And we have you are also told about the different types of non-coding RNA. What are the non-coding RNA that were told you yesterday? Anyone can tell me quickly?
miRNA.
miRNA and?
rRNA, tRNA.
Yeah, and tRNA, rRNA, sure. And? Along with this, there are also some RNAs which are called as the siRNA, silencing RNA it is called. And madam also asked you to find out what was the Nobel Prize given for about mRNA, what miRNA, about which year, and she asked you to find out some details. Did anyone venture to find out? No? Okay, so these are the small RNA, non-coding RNA, which are very important in the cell. There are many of them, like the siRNA, which is called as the silencing RNA. Then this microRNA is there, and then we have small nucleolar RNA, and many other varieties of the RNA. Many varieties of the RNA are there. Today we will just talk about a microRNA. Why microRNA is useful? Because these have approximately 22 nucleotides. Here you have to why I'm stressing on the number of nucleotides is there will be many number of my RNAs, small RNAs, non-coding RNAs in the cell, and which one is microRNA you have to identify if you don't know the type of RNA which is there. So if you want to know whether you are working with an miRNA or siRNA, then the length of the miRNA is important. So you need to remember that miRNA is a non-coding RNA of approximately 22 nucleotides. And why after so many non-coding RNA, we are in particular talking only about the miRNA. The reason is this miRNA will regulate the gene expression, And this miRNA, there is a collection of a database which is a collection of all the miRNAs which are going to be targets for different bioproteins. So these microRNAs, they activate or shut off the different pathways. So whenever any gene is switched off or any protein is not being synthesized, this could be possible that these microRNAs are eating up those mRNA and they are terminating the translation process. So this transcriptome, which is a collection of all the mRNA of the cell, it is called as a transcriptome. These are regulated by the miRNA. This is miRNA and transcriptome is mRNA, that is the messenger RNA. And these are conserved and serve as antisense regulators of the other RNA. So what is the major function of these microRNA? The major function of these microRNA is the downregulation of the protein function. They inhibit the protein function by inhibiting the translation of the protein from mRNA or by promoting degradation of mRNA. So how do you predict which which of these are the targets for these, which mRNA are the targets for this miRNA? So there are different approaches which can be done. One is called as a sequence-based approach, and another one is the conservation of 3' untranslated region and analysis of the microRNA and mRNA expression data. So these are the three tools or these strategies which you can use to identify the targets of the microRNA. So the increased expression of an miRNA specific to a target is associated with the decreased expression. So if a particular miRNA is overexpressed, you need to look from the data from its target, what are all the genes which it is going to switch them off, or which of the mRNA which are going to be turned off or switched off because of the presence of this miRNA. And yesterday when we were discussing, Madam also explained to you something regarding the stability of the RNA, thermodynamic stability, by showing the two different models, the centroid model and the MFA model, telling you that MFA model is a relaxed model and it can be having lesser energy, thermodynamic stability. So the analysis of thermodynamic stability of the microRNA mRNA complex also is important to study the effect of this microRNA. So this is just a brief idea of how this microRNA are going to help, or how the microRNA are regulating the translation of the desired gene product. As you can see here, you have this regular process of the transcription going on, then going on to the translation process, the ribosomes, and you have the ribosomes and the mRNA molecule. So this microRNA, if it is going to come and bind to this particular complex, which is a risk complex, it is going to cause the decay of mRNA, and when the decay of mRNA occurs, there is underexpression of the mRNA. So microRNA, they regulate the translation of the targeted gene product. One mechanism is by degradation of the mRNA. The second mechanism is blocking the initiation of the translation. And the next step is another step where they can regulate this process is via translocation to processing bodies. Why I have given this is make you understand why only mRNA or miRNA we are talking about, because it is the one that is directly related to the regulation of the gene expression. So what are the miRNA prediction databases? So we have to see what are the targets, identify the targets of the microRNA. So the miRNA base, the predicting miRNA target, that is the targetscan.org, and there is another database which is called as the mirdb.org. So MIR base, targetscan.org and mirdb.org. These are the three tools which I will be showing you today, where you will first identify the database of the miRNAs. First, you will look into the database of the miRNAs, and then after that, you will retrieve an mRNA sequence and predict what are the targets for that particular mRNA, what are all the miRNAs which are targets for that particular mRNA molecule. So that you can do by using two different databases. Okay, so just I will go through this process. This is predicting the secondary structure, which we'll not try to do now. Anyway, you can have the process with you. So you can select the species of interest and you select the miRNA from the display database and get the structure and retrieve the struct sequence of the miRNA. And then if you're wanting to predict, as I told you, we'll go to the targetscan.org. Okay, so let me go back to the Windows. Let me go back to, now I'll stop sharing this slide. I'll just share another process. Then we will come the step by step. We will go back to that and we will see. Okay. So now I will keep the open windows. I will also ask you, please keep these windows open. One is the MIR base and another is the target scan. Okay, so once the windows are open, then we can finish the work quickly. Okay, so here I will just share you the protocol once. We'll go by the protocol. I wanted to share another one, so this is not this one. Just give me a minute, something is wrong here. Is it sharing?
Yes, ma'am. Sheet is being shared.
Yeah, that's one.
Okay.
So this, I will just to show you this particular one, what you are supposed to do stepwise. First, you will open the MIR in MIR base and click on the browse tab. So let us go to MIR base. Go to your browser and select MIR base. Okay, so you will be getting this particular screen and then you select MIR base. So you will go to this location. This is the screen you will be getting. Next, after that, you select the species of interest, whatever species you want to see. Okay, so here you can see browse, click on the browse and you can go, for instance, if you want to check the human genome, human database, MIR database, go to the human thing, you will see a big list of miRNAs. So there are several of them which are there. This is the sheet which you would be getting there. This is the name of the RNA. This is the accession number. And this is the chromosome position. So here, importantly, students, how to know the chromosome position is, how to identify the miRNA is, these miRNAs, they should be having a sequence in the chromosome itself. Any RNA cannot be working as an miRNA. One of the conditions that the RNA is, you have to identify a particular RNA as miRNA is that it should be having a location on any particular chromosome. So that indicates that it is a real miRNA molecule. If you have an snRNA or any other type of RNA, it will not be interfering or it will not be giving you the correct result. So one of the conditions is that though this miRNA is only 22 nucleotides, its precursor, the precursor means the previous form of this miRNA, it should have some 80 to 90 nucleotides in length. From that, it is going to be processed and coming to 22 nucleotides. So that is one condition. And the next condition is that the miRNA, it should be localized on some position on the chromosome. So you know on which position the chromosomes, which chromosomes the miRNA position is located. If your gene is also located on that same chromosome, then you can see that that chromosome would be, that gene would have been maybe a target of your miRNA molecule. So we have these different types of miRNAs which are localized on different chromosomes. You can see some chromosome X are there, chromosome 13, chromosome 9, chromosome 21. So that is the location of the chromosome. So select the species of interest and then you will get the list is what we have seen. And if you want to know the details about a particular miRNA, now you can use all these are active links. So if you just click on any of them, it will be taking you to that page and the details about the miRNA molecule. So what is the details it is giving to you? It is giving the sequence of the miRNA here. You can see yesterday we were told the notation. What is the notation which is given for any RNA? You would have these brackets which are there. So what do these brackets indicate? The bracket positions indicate that these these positions are in linkage, which you can see here. Okay, these are the linkages which are possible here. UGG, you can see both these arrows are facing in the opposite directions. So wherever there is an opposite direction window brackets which are there, those sequences which are in a linkage with each other. And this sequence UAA, UAG, which is not having any commonality, that is in the loop structure. So totally this would be having a condition where you are satisfying that it would be having a definite number of 21 or 22 nucleotides, and then you can see other details, or you can go to other links what you have used yesterday, RFAM, and all those links which you can see. You can also put this in your structure prediction and predict the structure of the mRNA molecule. So this is the details of the MIR base, where you can see the location of the chromosome. And if you want to see this position, you can go to your genome browser. I had opened the genome browser. Yeah, if you go back to your genome browser here, you know how to get to the genome browser. It is the UCSC genome browser. So here, if you, what are the positions which we have seen? The chromosome positions we have seen in the previous case in the window. And anyone? Chromosome 9, I think we saw. Okay. So here, if you say chromosome 9, let us see that whether it will show
Padma
any.
ma'am,
Yeah.
you are showing the UCSC browser.
Yeah.
No, it is not been shared. Word document only is being shared till now.
What document is sharing? Just a minute, I will share. You can see UCSC browser.
Yes, ma'am. Now it is visible.
Okay. So here you can put chromosome 9. Let us see whether it works. And also you can visualize the tracks. This is only to make you understand that it is part of the chromosome sequence, and you would be having some miRNA regulatory sites and all that. Okay, so let us, so you can see this one, this particular list of sequences which are there. So the positions we can you can choose the tracks on this to see the number of miRNAs and other positions which are there in this chromosome 9. Okay, then coming back to our Word document is sharing, not sharing.
Sequences are in PPT.
Yeah, just I will share. Some issue with that sharing that window. Now it is sharing.
Yes, ma'am.
Oh, yeah. Okay, so we have completed this one because I'm running out of time. I'm moving fast. So complete data of the miRNA is given there. Then you can retrieve the sequence of the miRNA. Okay, so that is the way you can see on which position of the chromosome this miRNA is there, and you can see the sequence of the miRNA on the UCSC genome browser, provided you are taking a human genome or you are taking any of those genomes which are there on the browser. It may be any model organism whose genome is available on the browser. Now let us go to the next aspect, predicting the miRNA target. So predicting the miRNA target, you need to go to the target scan. I go to the target scan. You can go through the browser itself. This is this window of the target scan. You can see this is the human one, prediction of microRNA targets. What is the difference between this resource and the previous resource? The difference is there you are looking at the database of miRNAs. Now here you are going to take an mRNA sequence and predict the sequence, or you are going to see what are all the microRNA which are possible. So here you have an option, that is you can select a species, you can enter a gene symbol, or you can select any reason, or you can enter a microRNA name. So I'll go back to the PPT here. So this is a target scan which is there. So you can enter the miRNA number here. So when you are entering, you make sure that it is given in a particular format. That format has to be strictly adhered to. You cannot put it. You have to give this particular number like this, miRNA, and then it will be giving you the list of the miRNA. So you need to open the NCBI database, select any mRNA sequence, save the sequence, and then you can predict the sequence, right? Okay. So here I'm going back to this, you will be getting an output we have just seen like that, and from that you can select the list of the target genes. Okay, so I will quickly go through this today. You can work it out, and next class you can write down the steps and you can pass through this. And then on Monday when we join back, if you are unable to do it, then I would spend some more time and show you if my class is there, or any next class when I come over. In that class I can show you. Okay, not necessarily on Monday, but what I want you all to do is you please go through these steps. You make a note of this, and then go to your NCBI browser, take out the sequence, mRNA sequence, and put it through these different resources. So the other one is the miRDB site. This one also I will show you. So this is the microRNA target. Screen is sharing, right?
Yes, ma'am.
Yes.
So microRNA target prediction database. So this is the window where you can search by miRNA. You have this custom prediction. Go to custom prediction here. In the custom prediction, you select the custom prediction. You have other resources also. Target search is there. In target search, you can search by miRNA name or you can search by gene symbol here. You can give the gene symbol in this window. You can search by target expression or you can target mining is there. Select example, you can go through the multiple miRNAs or gene mining means you are trying to see from many miRNAs. The most comfortable one is your custom prediction. In custom prediction, you can just take the submission type, but here make sure you are choosing mRNA target sequence. And when you take the mRNA target sequence, you need to go back to the NCBI site. Go to the nucleotide. Okay. So select your mRNA sequence. Here, make sure you select an mRNA. Okay. So let us see where there is an mRNA here. There is an mRNA sequence here, but this is not human sequence. Let us set humans, Homo sapiens. So make sure if you are using that, this is because of human species, we will take human sequence only. So I will take this mRNA sequence, human sequence. And what is the format in which we are supposed to be pasting here? It doesn't ask. You can see the example here. You can look at the example, what is given here. So you don't require the FASTA sequence. So you can take the normal data. You need not take the entire sequence. So you can just start from the first nucleotide position. Okay, this is quite a big one. Okay, so you can take that and let me see if it works. Otherwise, you can try out. So this length is little longer. So make sure that your mRNA is little short. Some of these bases are not. Some of these are not accepted. This is linear DNA, linear mRNA. Let me try this one. So here, this is the limitation of this particular resource that you have to be sure the size is not big when you're working. Let us see if this works. This is also maybe here we have not selected mRNA. So here now it will work. So make sure that if you're loading mRNA sequence, you select the mRNA. So here retrieve the prediction result. So this will tell you it will give you the rank order, which is the score of this, which is the best possible score possible for that particular target. What we have seen, target details you can get. This is the miRNA sequence. Okay, custom target sequence is this one, for which it has designed the targets. And you can also look at this particular miRNA sequence here, the databases, and you can use this sequence to see the genomic location is given chromosome 6, and this is the MIR base ID, and this is the precursor sequence. As I told you, every miRNA, it should have a precursor sequence. This is the actual sequence of your mRNA, 21 nucleotides. But this is the, you can see UUUCAC, the red one which is highlighted is the miRNA sequence, but still there are other bases which are there. So this is the indication that you are actually looking at an miRNA and not any other RNA. So it should have the precursor sequence. The miRNA name is having 5P, but the precursor is only 7159. There is no 5P here. So this is the position of the miRNA on the chromosome. So you can go back to the genome browser on chromosome 6 and you can locate the position of the miRNA on the genome and see to which gene it is aligned or to which gene it is acting as a inhibitor or to which gene it is going to bind. Okay. Okay.
Yes, ma'am.
So you can try out the three resources: MIR based target scan and miRDB and yeah, we will share it because we are in the process of consolidating the different resource persons PPTs. Once this week's work is done, maybe next week we will try to share. Recording can be stopped, I think. Anita? Revati?
Yes, ma'am.
Recording is, you are doing the recording?
Padma,
No,
meere
ma'am.
chestunar, ma'am.
I don't have
Yeah.
the problem.